everybody, we are going to review for the upcoming test. Remember, if you have an A-day class, it's on Monday. If you have a B-day class, it is on Tuesday. All right, so first thing that you need to know about is the differences between DNA and RNA and some of the similarities too. So DNA is located in the nucleus. It has the sugar deoxyribose. It has two strands and it has the base thymine, which is different than RNA. RNA is located in the nucleus or the cytoplasm. It has the sugar ribose. It is single-stranded and it has the base uracil. Both DNA and RNA have phosphate groups in their backbone, and both DNA and RNA have adenine, guanine, and cytosine. So there are some similarities, some differences between the two. All right, so the transfer of information from DNA to a protein is kind of always in the same order for the majority of organisms. It goes from DNA to RNA to protein. So to get from DNA to RNA, that's the process of transcription. It happens in the nucleus, and your DNA is used as a template so that the RNA strand can be made, and that's mRNA that's being made. And then that mRNA strand moves out into the cytoplasm for translation to occur. So translation happens when that mRNA binds to a ribosome, and the ribosome reads the mRNA strand 3 letters at a time as those codons, and it calls over tRNA molecules with anticodons that match up with the mRNA strand. And that tRNA actually carries over amino acids, and they get all chained together to eventually build a protein. All right, so this is our codon wheel, and translation doesn't just start at the beginning of the mRNA strand. It actually starts when the ribosome finds the start codon, which is AUG. It codes for the methionine amino acid, and your ribosome won't start until it finds that codon. Your ribosome also won't stop until it finds a stop codon. So translation will keep happening until it reaches one of these 3 stop codons. On your codon wheel, you actually see the word stop right there. Those are the codons that code for a stop. And if you have a mutation to your DNA or your mRNA, that can cause a lot of errors if it messes up the starting or stopping codons. So we're going to talk about that in a little bit. All right, if you are still feeling uncomfortable using this codon wheel, we're going to quickly recap it, so hopefully we'll clear some stuff up. You need to always make sure that you're using it. Sorry, can't talk. Make sure that you're using it with your mRNA strand, not tRNA, not DNA. You're reading 3 letters at a time. Remember, those are your codons. And you use this wheel from the center out. So we're going to try it together with AUU, and then you're going to try it on your own with CGA. So for AUU, I'm going to the center of my wheel and finding A. Then within this quadrant, I go out to U and out once more to another U. And AUU, that codon, calls for the amino acid isoleucine. What I want you to do right now is pause this and try on your own to find CGA. Okay. Hopefully you found that CGA calls for the amino acid labeled right here, arginine. So I went from C to G to A, and that's how I found my amino acid. All right, next up are gene mutations. So there are 3 main gene mutations that we talked about: substitutions, insertions, and deletions. A substitution is when an incorrect base is put into your either your DNA strand or your mRNA strand. An insertion is when you add an extra base into the sequence, and a deletion is when you remove a base from the sequence. So if we look here, this top strand is regular, normal, unmutated DNA, the mRNA that it would then help to make, and the amino acids that would be made from that. In this substitution, this DNA was supposed to have a C, and it got mutated. Now it has a T. So that impacted my mRNA strand and 1 amino acid that was being called for. That's a substitution. This is an insertion right here. So we were supposed to have the codon CTT from our normal strand, but instead an extra C was added in. It was inserted into the sequence. So now we have a new codon CCT because it pushed everything over. And that impacted my amino acids from that point forward. Same thing goes with a deletion. We were supposed to have A, so 4 A's in a row. One of those A's was deleted out. It was removed from our sequence. And now all of my future amino acids have been impacted by that. That's called a frame shift mutation. So frame shift mutations are caused by insertions and deletions, and they change the reading frame. So it changes the way the the codons are grouped together. Remember, codons are read 3 letters at a time, and if an extra codon is added in or deleted out, that's going to change your grouping of 3 letters. If you look right here in this frame shift mutation, a uracil was deleted out of the sequence. So now my nitrogen bases have had to regroup their codons, and it had a really big impact on this amino acid chain that was being made because the new groupings actually put a stop codon in too early, and now translation has ended earlier than it should have, and your protein wasn't made properly. So frameshift mutations normally have a much bigger impact than substitutions. All right, hopefully this helped you feel a little bit more prepared for your test. Please make sure you reach out if you have any other questions. We are happy to help you, and I hope you have a nice weekend.